All right, so I'm really excited to have Brian Coventry with us tonight. So Brian did his PhD with David Baker at the University of Washington in Seattle, and now he's a research scientist also for the IPD. And Brian's work is focused on the one-sided de novo interface design problem. So you've got a structure of the target, you don't know what a binder should look like. How can you design a protein that binds to a given site? This has been something of a white whale for the protein design field, I would say, for a very long time now. And um, Brian, I would say, has made a remarkable amount of progress in this, more than the rest of the field combined. And together with some other colleagues at IPD, they just deposited a paper in bioRxiv, which proposes the first general solution, not a flag plant. We did it once, but as you'll see today, did it many times. And so I'll stop talking. Brian, please take it away.
All right, hello, thank you. Let's try to share some screen. All right, let's see if this works. Okay, cool. So does this look like it's presenting?
Yep.
Okay, great, cool. Hello, so yes, as Chris said, I am Brian Coventry. This is my first ever seminar, so that's maybe a good thing or a bad thing. But in any case, I'm here to tell you about my PhD work. So this is based a lot on my defense, but it's fine because it's also based on the paper we just put out. And so goal is to make it 45 minutes. You can ask questions during if you like. It'll probably slow it down. But yeah, and so anyways, yes, as Chris said a little bit about me, he pretty much said this exact slide. I was a grad student in David Baker's lab with the stated goal of solving the protein interface design challenge, which was almost like a sarcastically given PhD project because we didn't think it was going to work, but it did work, and with a lot of help to a colleague that I'll be talking a lot about. And now I still work there. I'm just a research scientist now. So we've got three things to talk about. First, we've got the background to motivate what's going on here. Then the story of Longxing and Brian, and then what we know now. All right, so the first thing is the specific problem we're trying to solve. And so, I'm sure everyone here knows what proteins are, but we see this picture a lot of the SARS-CoV-2 virus particle. And each of these red things here is the spike protein. And so here's just a blown up version of the spike protein. This is the open state for those that are very nitpicky about this. And so what we know about the spike protein is it binds to the ACE2 receptor. So here is maybe it just binds to ACE2. So here is ACE2, and this is how the virus starts getting into your cells. So the virus binds with the RBD right here, the receptor binding domain. It binds to ACE2, and this is how it gets into your cells. And here's just a close-up of the interactions going on here. But the thing is, antibodies also bind to this spot. And so we, people in the field have identified antibodies that neutralize the COVID virus. And one of the ways you can do this is you can bind to the receptor and block it. And here's that same interface showing, a different way to bind to it. And so, one of the questions we had was, well, if antibodies can bind to this site, can we bind to this site? All you need to do is make a binder. It should be straightforward. And yes, turns out we can make a binder. And I'll point out this was mostly Longxing, but we made the method and the Longxing made the binder. And here we have yet another thing that binds to the spike protein. So we have a paper on this. It came out several months ago. This thing binds with picomolar affinity. We've got a bunch of different variants of it. But all in all, these things neutralize the virus in exactly the same way that the antibodies do. So that's pretty cool. We were able to make something that neutralizes the COVID virus. And it's going through clinical trial. Well, it's not in clinical trials yet, but it's moving towards there. So maybe someday this will be a drug, but that's a slow process. But the main point to take away here is we have three very different proteins and three very different interfaces. And so the thing you can start to see here is that, back in the day, people studied interfaces, and there's the various ideas of hot spots, conserved binding loops and such, and certainly those things do matter. But, there's something deeper going on here. Perhaps there's some physical fundamentals underlying this that if we understood them, we can make any interface we wanted. And so how do we go about learning about them? Well, so historically, we've had these three options, really. You can either look at native interfaces, you can steal native interfaces, or you can start from scratch. There's probably a 2.5 in there somewhere. But so the problem here is if you study native interfaces, so a lot of people have done this. So unless you have new data, it might be replicate work. And the biggest thing that they've learned is that you really need to get shape complementarity and solvent accessible surface area. These are the two biggest things that you find when you look at this. And although, these things correlate, the question is there something else underlying these? And you're looking at correlatives to the real thing. It's also hard to say you fully understand it if you're not testing. I mean, sure, you can test it on holdouts, but you're not making new proteins. And so the other thing you could do is you can just take parts of native interfaces. So although this works, you definitely get binders, but you don't quite learn as much. So one of the things that you really start to figure out is you get really good at grafting parts of proteins, but that's not quite, understanding how interfaces work. You're getting good at grafting, you're not getting good at understanding how interfaces work, which brings us to our strategy, the totally de novo way. The problem here is it's really hard. And the other problem is if you fail, you don't have any feedback at all. Like all you know is that didn't work. You don't know if you were close, you don't know if you were far, and you don't have any feedback about why it didn't work. All you know is that it didn't. And so that leads us to the arbitrary challenge that we selected for my PhD, which is design totally de novo binders using no native information. So the inputs, again, arbitrarily chosen by us, are you take a crystal structure from the PDB, you delete everything that interacts with it, you forget that exists, and you just try to make a binder to whatever you have on your screen. And so, yeah, we're ignoring everything about the native complex. We're using totally de novo proteins because our lab was very into de novo proteins for a while. And again, just using only information we've gleaned from the past, nothing that we've gleaned from this structure in particular. And we're going to put the success criteria as whatever protein we make, you have to actually be able to express it. And like, it has to be like a protein you can use. Arbitrary binding cutoff of about 1 micromolar. I mean, maybe we'll accept a 5 micromolar, but we're not going to accept like 100 micromolar. And also, since we're trying to, de novo design proteins, if we get a crystal structure, it needs to pretty much match our design. Like if it binds in a totally new way in the crystal structure, that's a failure. Like, good job, you made a binder, but it was like, you didn't, I don't know. It's not the same thing. So this is the challenge. And so now we take us to the story of Longxing and Brian. All right, so the story starts in 2017. Back then, there was a very large binder experiment that had just failed. The experimental details are a little messy, but the current state-of-the-art methods were used against 7 different targets, and very few to 0 binders were identified. Again, experimental details. I was working on inserting loops into scaffolds, not a very promising way of going at the moment. Again, totally de novo loops, we still can't do that. So, and Longxing had just joined the lab. And at this point, we were working, again, with data from native proteins and grafting experiments. We didn't have the diverse set of data that we really need to kickstart the process. And so the first project was stopping the flu. So at that time, everyone really enjoyed making binders to the flu to try to stop it. And there's this conserved loop down here that if you can bind there, you can neutralize the virus. And there's various hemagglutinins with the flu, you know, H1N1 that specifies a hemagglutinin 1, neuraminidase 1. So H1 had been tackled already. We were trying to bind to H3. And we got our team here, Longxing and Eva. And so Eva was the one, well, Eva along with Chris and Gabe, they're on the next slide, but they were the ones pioneering this before I got there. And then Longxing and I showed up, and Longxing and I were a team that came at this together along with Eva. And we spent like 4 or 5 months making binders to this. We had like 30 parallel experiments lined up to figure out exactly the best way to make binders. So once we got the data, we'd know exactly what protocols to use, and it was going to be great. So we did the experiment and nothing worked. Okay. And so that's really sad because one, I mean, you know, 4 months of time wasted. And then again, with the nothing working concept, like we didn't really learn anything. All we know is it didn't work. Like, you know, those 30 parallel experiments, well, good job there because they all failed. And so that was kind of sad. And then just a quick step back, you might be asking, how do we test 15,000 binders? And I'm sure with the protein group, you guys know about this. But so as you know, we can synthesize DNA in a chip. We can order 100,000 sequences at once. You can transform those into yeast. We've got our cell sorter that can sort 5,000 yeast cells per second. And then we can thank the Human Genome Project for being able to sequence the DNA that came out. And just a little more specific, we call the invention of mini proteins. I asked Gabe Brocklin, and he said these are the 3 that put the mini proteins together. There's Chris Bahl. Thank you, Chris. And so the original plan here was to take these nucleotides, transform them into yeast, and get your protein on the surface of yeast. From there, I'm sure we're all familiar with Gabe Brocklin's work, where he used a protease to chew these up. And with that, determined some of the early foundations for making stable proteins. It's quite easy to repurpose the pipeline for binding. Instead of testing for protease stability, you label your target protein with another fluorophore, and then you're able to sort it with a two-channel sorter, with your x-axis being binding, x-axis being expression, y-axis being binding, and you can get your cells. And this is about as far as we're going to go into this. I'm not going to show you facts data, but this is where most of their high throughput data is coming from. And so back to protein design. So we figured, well, H3 is a hard target. You know, it's a viral target. The virus doesn't want you to stick to it. So obviously it's going to be hard. There's even like a glycan next to the site that knocks your binder off. So we picked another target, CD86. We thought, well, let's go after a human target, one that's friendly, spent several months making binders to it, tested them, and nothing worked. Which again, it's just really sad because, I mean, your slope is 0 here. So how many more of these do we have to do until something works? You really have no idea. And so then we could bring up the famous engineering cycle of design, test, learn. So this is what we want to be working on. But the problem is that you need binders to learn, and you need to learn to make binders. And so we'd love to use this process, but we can't because the cycle hasn't been started. We're somewhere beside the cycle and we can't jump into it. And so we had a few hypotheses as to what's wrong. I mean, the 2 simple ones are either you're designing bad interfaces or you're designing bad proteins. I mean, what else could be wrong, right? I mean, either you don't know how to make an interface or you don't know how to make a protein. And the tricky part is that if you don't know how to make a protein, you get the same readout as your interface being bad. So you just have to kind of guess which one's wrong and try to make it better. And then also at the time, we had the question, is this even possible? Because, I mean, you know, it hadn't been done yet. And with the success rate of 0, that's not great. And so, you know, the problems here are, so our interfaces are bad, but compared to what, right? Like our interfaces had the same metrics as native interfaces. It sort of means your metrics are wrong. And then maybe our proteins aren't folding correctly, but they all pass the protease assay. So they're all folding into they're all folding into something that's protease resistant. That's what I'll say about protease assay. And the thing is, you can't crystallize all your scaffolds. I mean, I've asked for this, but you can't crystallize 100 proteins, and even if you did, 100's a small fraction of what we have. So Longxing had the brilliant idea, let's just make things that look good. Okay, taking the bold move here that we as humans understand interfaces more than our metrics do, with the basic idea being that your metrics aren't good enough. Let's just do this brute force by hand. And just my shout out to Longxing here. Longxing once looked at 10,000 proteins in one day, filtering for a chip order. It's a record that's never been matched. But yeah, so our interfaces looked really bad. And so this is a schematic because it's really hard to show proteins in a PowerPoint presentation. But so the thing is, your native interface looks really good. Close contacts, good packing. And then our interface is like, these are rather accurate. Like you have proteins coming in at weird angles, making no interactions, just weird off angle. But the thing was, our metrics said that these were good. And we know they're not good. They look terrible. But so the metrics weren't there. And so we made a new metric. In the paper, this ended up being called contact molecular surface. It's a great metric. It's like a distance weighted surface area. But anyways, using this distance weighted surface area, which I call target proximity here, basically our goal was to make sure we're getting into all the nooks and crannies. And so using this new metric, we see the native interface checks all three boxes, but only one of our designs even kind of gets close to one of these. And so with this new metric in hand, we found out that most of our designs fail the new metric, which is a good thing. I mean, because that way we can actually do some filtering here, but we're talking like 1 out of 1,000. And so that's not great when you're trying to make something that only 1 out of 1,000 look good. And so the obvious thing to do is to upgrade your design protocol. And so here's what the design protocol looks like in general. There's a lot of ways to make binders to things, but this is the one that we've chosen. So what you do is you start off with your library of stable mini proteins. These were all verified by the protease assay, which guarantees they fold into something. Which I say that because at the time, people were really convinced if they passed the protease assay, they're atomically accurate, but I have some counter data to that. Anyways, you take your library of mini proteins, you dock them to the target in every way imaginable, and then you mutate your interface residues to make them so that they're actually binding proteins. And then there's a filtering step afterwards. And just schematically, I'll just show it like this so we can talk about the changes to it. You've got your docking, which we use a program called RiffDock, which I'll get into, interface design, and finally your filtering using your favorite metrics that you've developed. And so first we'll talk about how we made the design phase better. So let's hope this plays. Cool, yeah. So our interface design is using Rosetta. The general idea here is that you use Monte Carlo to randomly change the interface to try to improve the energetics. And then you follow that with minimization, as you can see happening there, to move the structure around a little bit. And so I only bring this up because one of the changes was to change how this works. In the original one, we go down to 2% of the repulsive weight, which let things get really close together. And then we slow, so basically the idea is you allow big amino acids to fit into places, and then you kind of increase the force so that things start to move out of the way and fit together. Yes. And so we made 2 changes to interface design here. The first one was, like I said, we gave Rosetta, well, actually this was Jack McGuire, but we gave Rosetta a better ramping schedule. It's a subtle change, but it made a huge difference. This probably only matters if you're running Rosetta. And then secondly, we told Rosetta that it needs to favor the interface. Like the problem is that by default, Rosetta tries to optimize the overall energy, but it's like, no, 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 focus on the interface. Don't worry about the scaffold. If the scaffold's bad, we'll throw it out. But it's better to have a good interface and a bad scaffold and throw that out than Rosetta never trying. Great, so that improved our interface design. Next up, we need to fix the docking phase. Yeah, so here's where RiffDoc comes in. RiffDoc's this neat program written by Will Scheffler. And the idea is you can pre-compute all of the good interactions with the target beforehand. And this is a tiny fraction of what's stored. And then you can store the backbone positions in a hash table, like a 6-dimensional hash table. That way, scoring is looking up into a table. It's not packing in like individual atomic scores. It's just look into a hash table, pull a number out. And so this is how we do the docking with the target. Here's an example of so then you can put your scaffold into the field, and for any given dock, you can very quickly ask, how good is this dock? Okay, and so originally the way this worked was that we have the RiffDoc protocol, and it used this thing called the hierarchical search that I don't have time to go into now, but it's just a branch and bound search using low resolution gridding of space. So instead of having the hash table be a ½ angstrom, you blow it up to 16 angstroms and you take the best tryptophan we found in 16 angstroms. And so the problem here is that the vast majority of the outputs look bad. I'm talking vast majority. Okay, it gets lost at the 16 angstrom phase, but it's pretty quick at least. So it only takes 1 second per output, and internally it's like 20 milliseconds per dock. This thing is fast, but yeah, the problem is the vast majority look bad. And so first thing that Longxing did was we stuck PatchDoc in front of it. PatchDoc's a program that like docks based on like very coarse shape complementarity. So you can imagine cartoon mode docking where it's just like, do these things fit together? We got it to output at about 3 angstroms. And then, so now only the majority of the RiffDoc outputs looked bad, which is a step forward. I mean, we went from almost all of them being bad to now only most of them being bad. But now there's a problem here. The problem is that RiffDoc's outputting bad outputs once every second, but the interface design takes 300 seconds. And so you'd like to bridge this gap and maybe only design things that look good and not design everything. And so that's where we bring in this thing I call the predictor, which unfortunately it's getting published under the name the predictor, which I kind of regret. But anyways, this thing takes 15 seconds and it can filter your RiffDoc output such that they look good. And I have a slide on this from my dissertation, so we'll show that real quick. So the way this works is you've got your RiffDoc outputs. You take a small pilot study of them and you subject them to a fast interface design as well as the real thing. So this is the slow one. This is some fast new thing I invented here. And then what you can do is you can put both of these into machine learning to say, okay, if the fast outputs looked like this, what are they going to look like after real interface design? And this is like, it's like logistic regression. It's not like deep learning. But anyways, the reason this is good is that these 6 million outputs don't need to go through the slow step. You put your 16 million outputs through the fast step and then use your machine learning thing to pick the best ones. And with this, we're able to greatly enrich our outputs for good designs, spending only 15 seconds per design. Great, so that's how we, yeah, so back to this, we have one last thing to improve. So you might think that after interface design, you're done and you filter. Well, we added an entire another step right here. And so what I'm going to talk about now is this idea of rare events. When you're doing protein design, sometimes you get outputs that are exceptionally good, way better than the rest. And the thing is, they're really rare. So here I've shown that only 1 of my 6 outputs made this great interaction. And you know, this could be like 1 out of 10,000 in protein design land. And so the thing is, you'd like to take advantage of the fact that sometimes you found great interactions. So we did this thing that we call motif graft or resampling, where what you do is you take your scaffold set and you superimpose them onto the good interaction. And you can see with the superposition method in this cartoon example, each thing we dock once and find a great dock. Whereas if we're just doing, two-dimensional random sampling here, you're going to explore a lot of bad docks and you might not even sample the good one. And so using this, we're able to, double down on anything that looks good and make a lot more of it. And just to show you what these look like in the real world, here's some motifs we extracted. I could have put side chains on these. They have side chains. But yeah, so these are what our motifs look like. They're just parts of helices. And so, yeah, so that gives us our final design protocol, which is way longer. We've got our docking phase with the patch dock plus rift dock method. The predictor narrows it down. Take that into interface design, where we extract our favorite motifs, graph those motifs, run the predictor again. This is sort of like a really fast docking procedure right here. Then you do your interface design and you filter. And that's the new design protocol. It works a lot better. I can tell you about that in a few slides. But don't forget about protein folding. Just because your interfaces look great, your scaffold has to fold. If your scaffold folds wrong, it doesn't matter how good your interface was because it didn't form in solution. So the first thing, this one was Longxing. Longxing heroically looked at all like 10,000 of our scaffolds, and he threw out anything that looked suspicious. Just things that aren't really packed together, helices going all different directions. And so I tried to find like computational metrics that would correlate with Longxing, which eventually did work. I was able to correlate with Longxing pretty well. And so with that, we threw away the bad scaffolds, but then we ended up with like almost nothing left, which is a good thing, but now you have to make more. And so I made this cool protocol here. This is just a way to make helical bundles really fast. There's another protocol called sewing that's similar. I'll say I co-invented, like I came to the same conclusion that they did from separate angles. But anyways, picking a bunch of helices and turns, you can assemble helical bundles. And this is notable because this was 6,000 times faster than the old method. 6,000 times, okay? So we went from it being hard to make scaffolds to like, I made like 25 million in a week. And you still have to filter them, but it's a great place to be when you have way too many scaffolds rather than way too few. And so with our new tools, we picked up two new targets, IL-7 receptor and TRK. And so Longxing and I, we made 15,000 binders for each target. And we're expecting a success rate of 0. I didn't think this was ever going to work, but miraculously we got binders. And so for IL-7 receptor, we got 40 binders, and for TRK, we got 7. Now 40 out of 15,000 is not great, but it's something. The cycle of engineering is going to start now. And here's an example of the best one we found. I think this was like 10-ish nanomolar. I don't think this is the crystal, but there is a crystal of it and it matches. I have a slide on that later. But what do we do with the 44 binders? Do we try to make a correlation with 44 data points? I wouldn't recommend doing that. Do you try to figure out why these 44 worked instead of the 15,000? That's an okay strategy, but 44 is kind of low and there's also a lot of noise in these experiments. So instead, we did another experiment. The other experiment I'm sure you've all heard about, it's site saturation mutagenesis. You make every mutation at every position, and then from this, you get deeper insight into your protein. So you can look at your protein, you can see which positions allow mutations, which positions are conserved. You can look at the graph, you can ask, hey, does this make sense? Yes or no. You can see if everything looks good. And so with this data, we had our first, what I'm going to call like good data. So that the high throughput data, things sort of always enrich on yeast display. It's kind of, you're not quite clear if they're real or not. But with the SSMs, we can go through position by position and ask, is this making sense? And for IL-7, half of them were right. For TRK, only one of them was right, which again, that's why you don't, if only half of them or like 14% are correct, you probably shouldn't look at like the big correlations. But anyways, we looked through these by hand, and this was great because for the first time ever, we had confirmation that these things work the way we think they do. Like there's a hole and you should fill it with a hydrophobic. the data suggests that, which is great because I mean, we think the same thing too. You should fill holes with hydrophobics. And yes, we finally saw it. And we tried to predict this using Rosetta. Rosetta did okay. And so on the whole, these things seem to work the way we think they do, which is a good sign. There's nothing spooky or magical here. And so for the next year, we just kept doing the same thing, making little improvements along the way. Here's some proteins we made binders for. Here's some pictures of those proteins. And yeah, things are going pretty well. But the problem was, although each target gave us a new set of data, you know, we were like testing hypotheses and making a little bit of progress, but we weren't making all that much progress. And it was a little sad, but I mean, you know, we're making binders. We're getting our 5 out of 100,000 binders. We're getting there. But so the final breakthrough was actually because I had to present at a conference. I decided that I was just gonna pool all the data together and see what came out of that. And I mean, it makes sense in hindsight, but if you pool all your data together, you get a lot better correlations than if you look at each one individually. And so with that, we move on to the final section, which is what we know now. All right, and so first, we have the binders to date slide. This was back when I presented my defense. We have more now, but I didn't want to remake this graph. And so on the whole, we have binders to a lot of targets. Our success rate's been kind of crappy, honestly. Well, it depends who you ask. If you ask me, I'm not happy with 0.1% success rate. Okay, here we got a 1%. There out here in the census work, I've gotten up to 9% before. But the thing is, I mean, 0.1% says you have some idea of what you're doing, but you don't understand it that well. And just to give the counter argument, you know, the true biologist would be like, you have 146 binders. We're gonna cure some diseases with this. So it depends what side of the coin you're on. I'm not happy with the success rate, but at the same time, we have a lot of binders. And then we run the SSM confirmation protocol on this. This just gives you an idea of what percentage of these ended up looking real, which on the whole is about half. So like half of half of the things that stick end up being correct. The orange bars are on different y-axis here. But so that's cool. Half of what you're making is real, but it also means half of what you're making is not real, which really makes the data analysis harder.
Hey, Brian?
Yeah.
So I have a question. What do you mean with is real? Like binds in the right spot or binds at all?
Yeah, binds in the right. Yeah. Binds in the right spot as well as we can tell from the data.
Okay, but the other was still binding. They're just not binding where you think they should bind.
The others fall into a few categories. There's the it didn't bind at all category. There's the something weird is going on with this protein, but it binds. And then there's the this is binding, but probably not for the right reason. There's like various levels of bad.
Okay, thanks.
Yeah, in terms of the ones that some appear, so it's pretty embarrassing when you order these things and they don't work. We've sort of figured that out. There's a lot of oddities in yeast display. You can get doubly transformed yeast cells, which make it exciting. But that's where a lot of the ones that don't bind come from. Yeah, but anyways, with the data pooled together, the biggest correlation that came out of here was target hydrophobicity. Okay, and so this x-axis here is it's a tool that I think more people should be aware of. It's called the SAP or SAP score. It came out of, I should have had a citation on here. It's the developability index is the paper that it's from. So if you search that, you'll find it. But it's a measure of exposed hydrophobicity. It uses the 3D model and SAS and the intrinsic hydrophobicity of each protein. But anyways, the bigger the number is, the more hydrophobic surface area that's exposed. And yes, this does work better than exposed hydrophobic surface area. But the biggest takeaway here is that there's a pretty strong correlation between hydrophobicity and success rate. So the very hydrophobic proteins, we made a lot of binders to. But the ones that weren't very hydrophobic, we don't have that many binders to. And again, since this has happened, I've continued to explore out in this area as the need for binders increases. And there's just a cutoff. Like at some point, you can't make binders. So polar binders are something that we're working on. In terms of the binder side, the binder hydrophobicity has a sweet spot. If you make an entirely polar binder, it doesn't work. If you make a really, really greasy binder, it doesn't work either. And so some hypotheses here at the low end, perhaps your binder just is more happy being a monomer than it is binding to something. At the high end, we're thinking these things form homodimers. So we sort of know a lot of our binders do form homodimers, but at the very high hydrophobicities, these homodimers might be so strong that the binder itself is outcompeting the target interaction. And so we think that's the best explanation for this trend. So other than hydrophobicity, the next most correlated thing is like packing and interface size. So these are the things people found studying native interfaces, right? That you want to have good packing, and you also want to have like, you know, bigger is better for the most part. And so we just have some metrics here. We've got, like I said, this is a contact molecular surface. It's a weighted surface area. It's actually like the code for this is stolen out of the Lewis and Coleman shape complementarity. Shape complementarity is a number you can calculate between 0 and 1. I've got my own opinions on this filter, but it does correlate fairly well on the data. Rosetta's predicted binding energy correlates as well. And so, I mean, again, these are all things you expect. It's nice to see that the data holds up to what you're expecting. It doesn't always. For instance, if instead you just use solvent accessible surface area, this correlation doesn't hold at all, because solvent accessible surface area is not a very robust metric. And then another correlation that really jumped out was net charge. And so this one took me a while to figure out. But so net charge here, we have, you know, if you're very negative, you can make binders. If you're very positive, you can make binders. And so the hypothesis here is that the hypothesis here is that we're breaking homodimers. And then negative and positive charges are interesting. Negatively charged proteins behave very well. They're the kind of proteins you want to work with. Positively charged proteins don't behave well. Positive charge gives you this huge success rate boost when you're trying to make binders. It's our method of last resort now. But the problem is these things don't express well. And I know we had one collaborator that found that our positively charged binders stuck to glass, which is just concerning. So negative charge is the way to go. They're they're more monodispersed, and they bind on each surface, which is great. Another correlation, the scaffold topology really matters. And so before we look at the weird ones, I want to draw your attention to the 3 and 4 helical bundles here, because these have very big denominators. Okay, so across all the targets with all 700,000 3 helical bundles, the success rate is like 2 to 3 times as high as 4 helical bundles. Now, there's a lot of explanations for why. So perhaps it's 65 amino acids. You just can't really form helices long enough to make a 4 helical bundle. With 4 helices, there's more ways for things to go wrong. There's a lot of ideas as to what's wrong here, but this is a very strong trend. And then the next trend is that the more beta sheets you have, the worse you are. And so this HEEHE topology is sort of an outlier. It kind of came from a weird experiment, so perhaps this isn't quite as robust as the others. But I mean, yeah, beta sheets are not in your favor if you're trying to design de novo proteins atomically accurate at the moment. But this still leads us to the question, why is the success rate so low? I mean, 0.2%. So we've got a few ideas, which the same ones from earlier. Maybe the interfaces are bad, maybe they're not folding correctly, or maybe they're getting trapped as homodimers, which we know this last one's kind of true, but it's also easy to get around by just not ordering super hydrophobic things and giving them a negative charge. So I came up with this really interesting scaffold accuracy test a while back when things weren't going well and I just wanted lots of data. And so the protocol here was I took some SSM validated interfaces. Now, these are not crystallized. They're just our design models that from the SSM looked correct. Oh, and I'll point out this was against IL-7 receptor, the first one. So I think we had 11 of these. And then what you do is you superimpose your scaffold library onto these helices, and then you just copy the interface side chains over. So really the question is how accurate does the scaffold need to be such that just copying the side chains over works? And so, we're like expecting to see a graph where you have like 100% success at the start, and then at some critical number, it drops to 0 or something. And here's that graph. And so what's interesting here is where the inflection point is. The inflection point is at like, what, 0.3, 0.4 angstroms RMSD? I thought it was going to be out here somewhere. Okay, so the numbers on top of here are the number of designs in each category. But I mean, I was really loading this up out here, hoping to capture the curve out here, but instead, just nothing out here. And so this graph indicates two things. One is that the required accuracy of your binder's backbone, okay, needs to be like definitely less than 1 angstrom RMSD, maybe as low as half angstrom. And two, the fact that we were even able to get this graph suggests our scaffolds are pretty accurate. Because if our scaffolds were only accurate to 1 angstrom RMSD on average, we couldn't have made this graph. The scaffolds have to be pretty accurate for us to actually get something that looks like this. So this sort of says that, your scaffolds might not be the most accurate, but they're probably not your biggest problem. And so the problem then must be that our interfaces are bad. And so, now we're arriving at state of the art here where we don't know why they're wrong. Maybe it's water. We don't consider water at all. Our electrostatics are terrible. We don't consider sidechain entropy or, backbone entropy or loop entropy. And we also don't consider the idea that perhaps there's a sequence of events that has to occur in order to produce a binder. I've seen lots of, one-off examples where that might be what's going on, but it's hard to say. And then lastly, and my slides are still set up in my dissertation mode, we're now switching to where I say, and then for all the biologists in the audience, here's the information to make you happy. Okay, and so first, the binders do work in purified form and they're hyperstable. So we have our biolayer interferometry here, the octet. I know people don't like octet and some of our graphs look pretty bad, but this is how we did it. CD, in this example, the thing doesn't melt at 95 degrees. There's the melting curve. Yeah, and then the next slide has a bunch of these. So here it is for a bunch of different targets. Yeah, I should have had the target name next to it. I apologize for not. Also, I know some of you in the audience are pretty upset about these octet plots. I apologize. We'll have to ask Longxing about that. Yeah, I mean, well, don't worry about the octet plots, but we have a lot of these. They all definitely bind on octet. Do they dissociate? I don't know. But anyways, from these, we fit KD values. That's what you should do with bad octet data. Here's some KD values based on that octet data. So take that with a grain of salt. But if you do take it with a grain of salt, we're ranging from picomolar up to micromolar. So that's cool with everything in between. It's definitely target dependent what numbers you get here. It's not so much method. It's like the target has a number you can achieve. It's if we redid these, maybe we can improve them a little bit, but it's the target.
A quick question: are any of these numbers after affinity maturation by SSM, or are these?
Thank you. They're all after that. Yeah, and I wish I had the data from before, but we never really titrate them before, unfortunately. That's a great point. Yes, these are. I skipped that. Yeah, the audience that knows what I'm talking about. Yes, we do combo optimize these at a later step, but it's in the paper. You can read about that. We have crystals. Here's a crystal of the trick A interface. I think in the paper we actually have 5 crystals now, so that'll be exciting. They're probably on the PDB if you look for them. But yeah, here's the crystal, and so then you're gonna then you're gonna ask the question, Brian, you just said that you have to have half angstrom accuracy. How come there's 1.4 angstrom accuracy here? Well, the answer is that, well, first of all, this binder kind of fell apart when it bound, so it's not the best example. But although the interface isn't super accurate, the designed binder is. And so here comes the COVID binder crystal, which has been out for a while. But so in all these circumstances, if you look at the RMSD of the interface of the binder to itself, the scaffold folded very accurately. The binding interface is exactly correct. But then interestingly, the whole binder shifts a little bit because Rosetta's not great at modeling this kind of thing. And so we have accurately designed proteins that then bind differently. But yeah, here's another crystal, side chains are accurate. And then we've got our great, diagonal binding curve here. Okay, and then since we have this audience, the reviewers don't like this graph. The y-axis is how high the trace went on the octet machine. Okay, so black here, black down the diagonal. These are all the interactions that we expected to work. So the flu binder. Oh, okay. Our flu binder binds to both H1 and H3. It's a pan flu binder, but the trick A binder binds to trick A. This target protein was kind of sticky, and so was this one. But in this crude assay here, we have mostly on target binding. And so you're thinking, great, the proteins you pulled out that bind specifically, but you sort 100,000 proteins at once. How specific is that library? And the answer there is it depends. And so it depends on how successful you are. So this is a library where I had 100,000 binders in the library targeted to 4 different proteins. And so the proteins are down there. And what you can see is for PDL1 here, where we actually had a decent success rate, the on target to off target was very striking. We're definitely making specific things here, and we're not just like phage displaying out of our library. However, for things where the success rate isn't so great, maybe you can make the sort of phage display argument. But what I will say here is we followed up on these with SSMs, and I'm very confident these are binding correctly. The VEGF ones actually were all bad, but these ones definitely are binding correctly. So although our success rate wasn't great, when we got the binders, they were correct. And so in conclusion, target hydrophobicity is the main driver. You have to have good packing, and yeah, in both the core of your binder as well as your interface if you want these to work. Bigger interfaces are better. Our success rate ranges from 0.1% to 1% for targets with specific criteria, which is mostly hydrophobicity. And then finally, if possible, you can do this. You can start with just a crystal structure and make a binder to it, which is pretty cool. And then lastly, we have the giant acknowledgement slide, which I want to thank Longxing, and that's why I have this picture right here. So David Baker, my boss, this is like his favorite picture. Longxing flew home with me to Ohio, and we took him to my family's Thanksgiving. So here's Longxing along with my extended family in Ohio at his first ever Thanksgiving. And with that, I will accept your questions.
That's a great talk, Brian. Thanks. That's really exciting. Let's open it up. Questions for Brian.
Can I ask the first question if I have to go? Awesome, Brian. Thank you so much for your talk. I had a question and a comment. So my question was with regards to the homodimer hypothesis that you brought up. So I'm not a yeast surface display expert, but my understanding of how the system works is that the attachment to alga-2 and bringing the protein to the surface would actually helps a lot in preventing those homodimers from forming. So at least in the yeast surface display assay, you probably wouldn't need to worry too much about those homodimer formation and like aggregation. And maybe that's wrong, but I was just wondering what your thoughts are on that. And then the comment was that in terms of like checking the specificity of your library, this is just like very philosophical at this point, but I feel like there are some targets that, depending on the amount of surface that you have exposed, you wouldn't expect the library to be super like specific, you know, like there are always parts of things that can bind to other things. So I don't know. But thank you so much. This is great talk.
Yeah, great. Yeah, thanks for the questions. Yeah, no, I totally agree that you can't always expect specificity, but I think it's important to show that sometimes you do have specificity. That way, it's not just the same library works for every target. But you're about the homodimers on yeast surface. I think it would. I don't know the specific papers and whatever, but I know someone, I think at RosettaCon, did some project that required a homodimer to form on yeast surface, and their thing worked. So I believe homodimers can form on yeast surface, but I mean, open question, so.
Thank you.
Maybe I'll follow up on Paris's question. Your off-target binders that you identified just right at the end of the talk here, back a slide or two, on the right side, the off-target. Did the same ones show up multiple times for different targets, or did you get a different selection of off-target binders in the library with each target?
It's a great question. They're different. Yeah, so it's not the same 22 binding all over the place. No, so I mean, those things probably are specific binders. They're just, you know, what is their structure look like? I don't know.
Cool.
I have a question. I was wondering, and seconding thanks for the talk. But I was wondering, when you chose PatchDoc to go before RiffDoc, what were your considerations in terms of choosing that compared to like ClustPro or HDoc or whatever else is out there?
So that wasn't me that picked it. So I can't.
Okay.
No, but it's fine. It's fine. I can give some pros of PatchDoc. PatchDoc was just in the lab around for a while. I'm not really sure why, but I'll say the biggest benefit of Patch. So okay, I don't know the other programs. I apologize, but the biggest benefit of PatchDoc is that it takes like 45 seconds.
Okay.
So, I mean, it doesn't need to be that fast, but it only takes 45 seconds. So it's definitely a plus.
That's very cool. Thank you.
I can fill in some history. PatchDoc was how the original binder designs for H1 that Cyril Fleischmann did way back in the day. That's how that was done.
Cool.
It's also great because you can convince it to ignore your side chains, which is what we wanted it for, which is just like cartoon level docking of proteins together. So.
Oh, that's really cool. Yeah.
Hi, Brian, I have a question. I was wondering about the delta SAP score, and if you had prospectively tried to find targets that had a very high value of that to see, to guide your selection of future targets.
Yes, certainly. So part of the Longxing and Brian story that I didn't tell is that when things weren't working, we went and tried to find the most hydrophobic targets that we could get our hands on. Okay, with the idea of let's just get something that sticks. And yeah, so we certainly have been, we certainly went through, and I would say that we've made binders to all the low-hanging fruit at this point. Now it's much more of a, upper IPD says, Brian, we need binders to this thing. I look at it and I give him the yes or no on it. That's kind of where we're at now.
What's the turnaround time if you had a new target to, I guess there's all these factors about experiments, but like, beginning to end, what's the workflow looking like to get to binders from the target?
Sure. So I'll give it to you as if you've got some high priority target that you need binders to right now, right? So you can get the well, it depends how many CPUs you have, right? We'll say the computation can be done in 1 to 2 weeks if you're just pushing it and you got a lot of CPUs behind you. We'll call a lot of CPUs 10,000. Okay, if you have 10,000 CPUs, you can get it done in 2 weeks. And then the wet lab time, I mean, again, this depends if you can get the vendors to work with you and get stuff to you faster. I would say a typical wet lab time is a month and a half. But if you somehow get priority, you could probably do it in like 3 weeks, maybe 4.
That sounds impressive.
But then I'll give you the caveat, you have to have a crystal structure. So new emerging target, you better have a crystal first.
I have every crystal structure now. I have the apo-fold.
Well, yeah, you know, that it would probably work. We haven't tried it, but it'd probably work.
Brian, I have a quick question. So far you've been trying to tackle 2 problems, like the de novo protein itself, and then trying to get it to bind to the interface. What if you use like non-de novo proteins, like try to repurpose existing scaffolds? I guess that's what people have sort of traditionally done. Do you think that would have increased the success rate, or do you really need the de novo designs, like to really match into the pocket?
Yeah, it would have. It's a different challenge. It probably would have helped the success rate if we were using the right native scaffolds. So the thing that's on your side here is that they have crystal structures. The thing that's not on your side is that they're more fragile. And so if you have a set of scaffolds that you're fairly confident you can mutate to whatever and they're going to work, your success rate would be much better. But if you're not confident that your mutations are going to leave the things intact, then your success rate might be lower, because our de novo proteins have the benefit that they're so rock solid that they can handle like 6 phenylalanines on their surface and still fold.
But would you say it's because it's rock solid, or is it because with de novo you have finer control of the, let's say, the interface? Like, is there something special about being able to arrange things? I guess, I mean, I guess that's what you would hope for, but maybe I could rephrase the question. So like, for example, the cases that did work, and let's say if you look for that particular interface somewhere, like just at the side of the design across all the PDBs out there, could you have found that somewhere? I don't know if you've done that experiment yet or not.
I see what you're saying. You're saying try to find the interface you're trying to design somewhere else in nature.
Yeah, yeah.
I don't think it would exist, because a lot of ours are just really, I mean, they're just, I don't know. Well, I mean, maybe we haven't tried, but my gut instinct is no.
Right. Thank you, Brian.
Yep.
Hello. I had a question. Did you notice a correlation between disorder at the target binding site and success rate of your designed binder?
Yeah, so I didn't show that here, but so this correlation, you know, on my computer has more data points on it, and there are some surprising failures. And I had a group meeting where I tried to figure out what was going on with surprising failures. It's like, this is really hydrophobic, but it didn't work. And yeah, one of the things that pops up is like, you tried to bind to loops. Like, and so yeah, we require hydrophobic residues that are well ordered. So like beta sheets, helices, great. Random loop, bad.
Thank you.
I was wondering, kind of also related to why things are working better than others. I wasn't clear. You're binding not necessarily to natural interfaces, natural binding sites, right?
I have an interesting answer here.
Okay.
For everything I said here in the paper we're putting out, yes, we ended up binding to all natural binding sites. However, in data that's not published yet, we bound to not natural binding sites.
Would you say that it's easier to bind to the natural binding sites?
It definitely is, and most likely the reason is that they're, you know, some sort of like kinetic kind of biophysical thing is better about those sites.
But all of the data you showed today, like all these things are basically on natural interfaces. It
is unfortunate, but it is, yes. However, our binding modes are totally different with totally new hotspots.
Yeah, I guess kind of what's interesting about that is like, is there something special about those sites, regardless of whether or not is there something special about that site that makes it attractive for building a protein-protein interface, regardless of whether you're recapitulating that interface? So if I'm understanding correctly, it does seem like you're more likely to get that binding if you're targeting a natural site, even if it's an irrelevant.
Yes, I would say that there is some other metric that I don't know about that looks a lot like this, where it's some metric I don't know, but native binding sites all score very well on it, and then other parts of the protein on average don't score as well.
Okay, great, thanks.
Hi, Brian. Well, thank you for your talk. It was so interesting. I would like to know what steps in your pipeline do you think you can still improve or you can optimize in the future?
Yeah, so it's a tricky question because of how broadly I overviewed this. When I gave this presentation for the first time, I'd say good luck. This is all pretty solid. The most obvious one to improve is the filtering step. I mean, like you'd figure with the right filters, we could get a 100% success rate, or at least, like 10%. So filtering, number one priority. So if I go back to, let's go back to the biggest picture I have of the thing. Okay, so here's the overall process. And what I'll tell you is when we actually go to place the order, okay, so right here at this bar, we get a bunch of designed proteins out. And right here, we also get a bunch of designed proteins out. And if you pool these together, like 90% of the good things are coming out of over here. So it's as if you don't need to make these proteins. And this entire upfront step right here is just a motif creation protocol, like a horribly inefficient motif creation protocol. And so if you could somehow just pull these motifs out of thin air, one of our reviewers mentioned the terms, the 2016 paper, or just maybe dock helices, you could probably skip this entire step and get a huge speed up here. And then from this is a pretty solid area. And then the interface design, I'm sure, can be improved. I mean, Rosetta's not the best. Rosetta's the best force field out there for this, but it's not a perfect force field.
Okay, thank you.
Hey, Brian.
Hello.
I had a couple of questions. So, so all the targets that I saw on your on the screen, the ones that I could recognize at least, all of them were extracellular targets.
They were, yes.
Have you tried any intracellular targets that you've validated yet?
I don't think we have. And the biggest reason is that we're concerned with what happens after we make these binders. And at the moment, we don't have a great way to get stuff into cells.
No. Okay, so my next question was a follow-up of that. And I guess it's generally for everybody working in the de novo area. Do you worry about immunogenicity? So if these de novo proteins are taken up by the cells?
So we do worry about that. And I can tell you a bit of data. So that original H1 flu binder, it's like not immunogenic at all somehow. So that our one data point is not immunogenic, but we certainly do worry about it, but we're not actively trying to fix that yet. However, none of our stuff have gone to the clinic. So perhaps if you come back to me 5 years from now, I'll say that was a huge mistake not worrying about it. So.
Got it. And I have one more question if there is time. So you said that there was a good correlation with DDG, which is very interesting because whenever we've tried to look at DDGs across different protein complexes, we've never quite been able to correlate it to binding energy or anything like that. So could you go back to the slide where you had the DDG correlation?
Yeah, and I have my reason why it's not working for you too. Yeah, this one. There it is.
Yeah.
Okay, so I have my answer. First, okay, so so one of the things that you learn when this is your PhD project is you learn what data you can compare and what data you can't compare. And although I lumped all the DDGs together here, that's a bad idea. The correlations work best per target. And so I didn't blow you guys away with like 20, ROC curves. I showed it like this, but it works way better on a per target basis. And also the other reason this works for me and not for you is that my proteins are all the same size.
Right. That makes a lot of sense.
For a 65 amino acid protein, a DDG of -90 is like very hard to achieve. But if your binding partners can grow arbitrary sizes, you're not going to see a correlation.
That makes sense. Thank you.
Yep.
Can you actually go back to this slide where you train the regression for
Yeah.
the prediction, the predictor, I guess?
Yeah.
Right? Can you elaborate a little bit on that? Have you looked into what the regression factors were, the main ones? Or
Yeah, I have. So this is a thing that started out as a very, very bold idea and ended up very simple. This machine learning, so everything on the slide stayed the same since the start, except for what this actually is. I started off with a random forest, okay? The problem there is that if you, our computational success rate of things out of here that end up looking good is really low, like 1 out of 1,000 kind of range. And so the thing is, if you want to train a random forest on 1 out of 1,000, you can't, you need a lot of examples. Like you probably need at least 200 examples, which means getting 200,000 docs, at which point, why are you even doing it if you're not saving any time? And so I tried to find some way that, one option is train your random forest on a bunch of data, but then you run into issues of is this still applicable and whatever. So what I ended up doing was, you can pretend, it's kind of true. It's kind of true that the individual metrics that you care about are independent. So DDG, how well you're contacting hydrophobic residues, shape complementarity, don't filter on shape complementarity. And so you, SASA, you pick all of your individual metrics and you do one-to-one correlations on them. So you basically say, okay, so the way this actually works, I don't have a slide on it, but you take your fast interface design outputs, and let's say they have DDG values ranging from something to something, okay? So you bin them into 10 groups, okay? Then if you have some final cutoff you're interested in, let's say you want a DDG of -60 kcal, according to Rosetta, you take each bin and you ask what fraction of the design in that bin passed this threshold. And if you do that, interestingly enough, you get a sigmoid out. And so what I then do is I fit an actual sigmoid to the data so that I avoid overfitting, because it's hard to overfit a sigmoid. And so then what I have is, for each metric that I care about, I have a sigmoid relating, if your score is this, here's the probability that you'll pass the filter. And so then what you can do is you can take all the metrics you care about with your sigmoids, and then for each output down here, you put them on each sigmoid, read off your probability values, multiply those together, and that's your final value. It has the same functional form as logistic regression. However, I obtain my weights independently. There's the predictor.
Nice.
Awesome. Thank you. Yeah.
Follow-up question while we're still on this. The fast interface design protocol, is that just pack rotamers with soft rep, or is there something?
I've tried. Okay, so my predictor script's on version 14 now. What this is, so this is what's worked best. You take the Rosetta score function and you start turning stuff off. okay, what actually ends up being left is like Leonard Jones, the implicit solvation, but specifically not LK-Bahl. LK-Bahl is really slow. I turn off all the intra-residue collision terms. I turn off FA-elect. I turn off hydrogen bonds, okay? And what this thing does is that makes the score function like 30 times faster. Then I greatly reduce the number of rotamers that are allowed to be built, and I restrict the amino acid choices. And that gets the pack rotamers mover down to 3 seconds. Okay, then you spend 7 seconds minimizing it. And so it's full rep pack rotamers, something like 1/3 rep on your first minimization, follow that with the full rep. And although the things you get out of this have a lot of alanines and look terrible, this is the most correlative. Yeah. Also, you can skip minimization if you don't want DDG. All the other metrics don't require minimization, but DDG does require minimization.
Awesome. Thanks, Brian.
There will be a non-empirical solution to this problem some point in our lives, like less parameter manipulation. Not anything wrong with that. Like that might be the solution. I think that some ways machine learning is just a lot of parameters, and that's highly successful. But is there, it's more of a philosophical question.
Well, I mean, this whole thing is a solution to an arbitrary problem that didn't need to exist, right? I mean, this is a solution to the fact that most of the RiffDoc outputs are bad. So like make RiffDoc better. I don't know. I mean, like, or replace this entire section with some deep learning thing. I don't know. But it's a quick way to reduce your doc outputs.
All right, well, if no more questions, let's thank Brian one more time for a really awesome seminar and great Q&A session.